welcome to week 7. How was Wales? Yeah, you all look incredibly relaxed. Got a lot of sunshine there, yeah? Yeah, really? Unbelievable. Did you see some posters of our ex-students? Yeah? Who?
I can't remember her name, but it was music and Cochlea and stuff.
Oh, right, one of Rachel's. Oh, excellent. Good. Fantastic. That could be you next year, by the way, now, yeah? Our students usually have either 1 year or 2 years later on their projects there, so that's good. Good. Right, what was I going to say? For next week, Mark and I have switched lectures, so it doesn't really matter very much for you, I suppose. But I will teach on Tuesday, the 22nd, and Mark will do this lecture on the 21st. And similarly, on December the 5th, Mark will lecture here, and I will lecture on the 6th in Building 3. Is that right? So not in this. Yeah? Where is that Building 3 actually? I'll find. I'm sure I will find it. It's just that I hope you will know where it is.
We were looking for these lectures and it said within 3 and we were within 7. Within 3 is not anymore.
It's not? Yeah. Oh. All right, I'll find it. You found it, so it must be doable. Okay, right. We are working our way up the auditory pathway. Last week we spoke about the basilar membrane and we spoke about how neurons code spikes or electric potential, action potentials. And this week we will put that into practice and we'll talk about the auditory nerve. So sound has gone up all the way now through the basilar membrane and we now investigate how the sound is actually coded neurally. Is that better or is it too? Maybe a little bit? A little bit. So how the auditory nerve works, anatomically and physiologically, what is the sound actually doing in the auditory nerve? How does it squeeze through the auditory nerve? We talk about 3 kinds of codings, of frequency, amplitude, and to understand that, a little bit about the Woolley principle. Okay, just as a reminder, little video, the basilar membrane of how it's vibrating. There's no sound here, there should be a little bit of sound. Okay, there's basilar membrane, tectorial membrane, inner hair cells, outer hair cells. You remember the outer hair cells are pulling a little bit on the tectorial membrane, but we're now interested in the inner hair cells, here blown up, and the stereocilia are floating within the liquid, within the perilymph, in the endolymph, sorry, in the scala media. And you see what I'm pointing out now here is that every time that it moves, not every time, from time to time, these become yellow. That means there is a spike happening, and it is so fast that it just rushes through the whole thing. It's so fast you can't see how fast it is actually going. And there are a couple of them, in fact around 20 of these on the inner hair cells. So that's the mechanics. Let's leave the mechanics now and go to the electrics of the membrane potential. Namely, what happens, actually if the sound was on, then you would hear every time that these become yellow, you would hear a little click. That's the spike that we can hear when we put it actually to a loudspeaker. Right, these are the stereocilia, and this is in vitro, so, but it's a very realistic situation because somehow we open the ion channels and the stereocilia, potassium flows in, and then as a result we observe the membrane potential. So the membrane potential fluctuates a little bit, but when they open it goes up. So we start here at minus 45, minus 50 millivolts, and it goes up to almost 0 up here. And as a result, you see this, that's actually without sound hard to see. So without stimulation they will respond from time to time, but when you stimulate them they will respond much more frequent. That's the point.
Perhaps you should increase the volume. It has about
No, it doesn't work because I understand why. Because I've got a microphone plugged into USB, and that means that the sound only goes through the microphone. I haven't got external speakers on here, and I want to leave the microphone in because it records the lecture. So it sounds like, without stimulation now, clack 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 clack, and now it comes clack 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 clack. Okay, I have to imagine that. Okay, that's what's called spontaneous activity. So without stimulation, when we're hovering around our membrane, our basic membrane potential, we have some stimulation or some spikes, a few. It's called random or spontaneous activity. Find out in a second what that's for. Okay, the other experiment, very simple, is thought experiment. We put our inner hair cell and we actually turn them around to the other side. So this is our situation where we have a little bit of random spontaneous activity, and if we close the ion channels on the stereocilia, you see that the membrane potentially actually decreases even further, and then the hair cell becomes silent. It mutes. So now it's random, and now it's nothing happening, nothing happening, nothing happening, and now it starts responding spontaneously again. So it's like a little, like a working point. You've got depending on how far your stereocilia are bent, you have in the middle a few spikes. If it goes to the right, you've got more spikes. If you go to the left, you've got less spikes. Okay, let's put that a little bit more scientific. How was it researched? Because it's interesting. In the 1940s, that already was started to investigate it by a guy called Galambos and Davies. These were the grandfathers of electrophysiology. That's one of the first sensory systems where electrophysiology was actually put into practice. We talked last week about the electrophysiology in the squid axon. That happened a few years earlier. But in a realistic, in a mammal, and in a sensory environment, 1943 is one of the very first recording. What they did is they put their electrodes within single nerve fibers, and I think they did it in guinea pigs, but I'm not quite sure about that. That is quite a difficult procedure because you need to open the head, you need to put a little microelectrode into the auditory nerve, which is just a millimeter or so across, and it contains a lot of nerve fibers, so you must be able to actually pick into one of these nerve fibers. I've done that a few years of my life, so I know how difficult it is. Nowadays we've got much better technology as well, because we see it in the microscope at the same time, which is not necessarily something that they could do. Right, what they did is they recorded increase in rate of firing to tonal and click stimuli. That's how they knew that they were in the auditory system. That's not necessarily something that is obvious if you're doing that for the first time. But it must have been very satisfying to actually realize that an auditory stimulus is somehow represented in the firing rate of the auditory nerve. That was then, of course, subsequently confirmed. Tasaki Kiang, 1965, he's the guy who did most of the work. Also related to the work of Bekishi on the mechanical tuning of the basilar membrane. So now, over the years, it was found that the mechanical tuning is actually represented somehow in the electric tuning of the nerve fibers. Horseradish peroxidase is a technique that we'll talk about a little bit more in the coming lectures, because this is what reveals the morphology of the neurons, because you can label neurons individually and you see where they're projecting to. So this was how it was discovered that the auditory nerve actually projects to the cochlear nucleus, we'll talk about next week. And all of that work was carried out solely on this type 1 auditory fibers innervating the inner hair cells. Why is it called type 1? I have that on the next slide. But that must be the quiz, which doesn't work, so I want to show of hands, which is slightly less anonymous. How many auditory nerve fibers do we humans have approximately, you reckon? Hands up for 3,000? 5,000? 1? 10,000? No? 30,000? 2, 3? 100,000? Most people, 2 million? No. The answer is 30,000. not 100,000. 30,000 is, well, you're not far off, you're just a factor of 2.5 out or something. But 30,000 is a take-home message, because it's not very many. 100,000 seems a lot, 30,000 is actually not very many, but yeah, enough. I put the number of 2 million on there because they have another justification, because that's the number of visual nerve fibers. Visual nerve, ocular nerve, is much, much thicker, so by a factor of almost thousands bigger. Okay, that's the innervation of the cochlea, and that's where it explains the inner hair cells and the outer hair cells, and the type 1 and type 2. That's what I want to refer to here. Right, you see here the one line of inner hair cells and 3 lines of outer hair cells. Okay, these are the guys that have the activity towards the brain. These outer hair cells get innervation from the brain. The actual nerve fibers that are connected to these are different, and you can see the difference even under the microscope. These are called type 1, these are called type 2, and these blue ones make up, they're not really blue, they're just colored blue here, they make up 95% of the fibers that go through the auditory nerve. They're also called bipolar or inner radial cells. That's bipolar as explained here. Why they're called bipolar? That's the way that they are set up. They've got a dendrite which sits on the inner hair cells, and we've got about 20 of these on each inner hair cell. So 20 different ones share one inner hair cell. Then the axon is extended from the dendrite to the terminal buttons, so to the synapses on the other side, and goes right through the soma. So it's quite different from the kind of neuron that I told you about last week as the standard neuron, but all neurons look somehow different, but they all share similar or similarities, and the similarity here is obviously that we have a soma, but we don't have many dendrites that summarize or that sum up the stimulation, but we only have one dendrite, and each time that will come above threshold, it will generate an axon, an action potential in the axon here, which rushes down through the soma, completely ignoring it, towards the terminal buttons inside the brain. Okay, this length here is a few millimeters. It's actually a few centimeters even, depending on your animal, because these happen in the brain stem. This happens in the cochlea, all of that. So actually you should make maybe 2 lines in there and say indicating this is a lot longer, obviously. Right, what's called bipolar, because we have soma has 2 poles, north and south, and they're both axons. The outer hair cells, or the outer radial, are unipolar cells. They look slightly different. The soma is connected to the axon, but you see that they've got many dendrites, they've got 1 axon, and they terminate into many terminal buttons again. Why that difference, I don't actually know. That's not necessarily a useful, I don't know what the use of this is. What else is to say about that? These are narrow, these are large, they are myelinated, which indicates they are fast, they carry the information with a precise temporal, high temporal precision. They are unmyelinated, so they are very slow compared, which also says that they are very much thinner. You see that they're thinner than these ones. And one auditory nerve fiber is directly connected to exactly 1 inner hair cell. 1 of these blue dots, 1 inner hair cell, but each inner hair cell has about 20 of these connected. The outer hair cells can go to many, so the outer hair cells go up here, and then they go to about 10 to 20 auditory outer hair cells, and one outer hair cell can support up to 6 of these auditory nerve fibers. Okay, so for us important ones obviously are the type 1, the inner, well, the inner radial ones that connect to the inner hair cells. And a little bit more anatomy. Put them both on. The outer hair cells have actually what wasn't shown on the previous slide. They have green and red connections. The green one are the afferent, and the green ones are the afferent. The red ones are the efferent neurons. The green ones weren't on the last slide, and there are just so few of them that we usually tend to ignore them. We don't even really know where they're terminating in the cochlear nucleus. This number 1 here is the cochlear nucleus, where all of our processing starts, where the neural processing starts, the first processing station of auditory perception. As you can see in the inner hair cells, the blue ones, this is where they all terminate here in the cochlear nucleus. The inner hair cells have a very, very few that are ignored on the last slide, efferent fibers as well, which we are not talking about, but we do talk about the efferent fibers, the red ones here, that goes to the outer hair cells, and to show you where they come from the contralateral and the ipsilateral, sorry, this is the ipsilateral, this is the contralateral superior olivary complexes, which are a completely different processing station and quite high up, if you like. So there's a lot of that has happened at this stage, and we'll spend some time in a lecture or two to talk about what these things actually do to the outer hair cells. But here we're concentrating just on the blue ones that go to the cochlear nucleus. So all afferent auditory nerve fibers terminate in the cochlear nucleus. The efferent ones come from the olivary complexes, from the ipsilateral, contralateral olivary complexes. Okay. Some information: the auditory nerve is primary auditory afferents. The fibers form the cochlear division of the 8th nerve. What's the other division? The vestibular, which we're not talking about. But I don't actually know how much, but it is about 80 or 90% of the cochlear nerve, I would say, is the auditory part of it. There's not that much vestibular nerve in terms of numbers. The cell bodies are called, or form what's called the spiral what's called the Rosenthal's canal within the modiolus. The cell bodies are within the spiral ganglion, within the cochlea. I should have a picture of the cochlea here. If you go back to lectures, you can find the spiral ganglion. It's within, not within the cochlea where it comes up, but just to the branches, if you like, the branches of the tree, before they then branch to the hair cells. They terminate in the cochlear nucleus, only ipsilateral. That's worth noting. All of these cells, all of these neurons, either apart from the red ones, efferent ones on the outer hair cells, terminate only ipsilateral and not contralateral. And we've got about 30,000 of these in each ear. The number, by the way, is about 3,000 inner hair cells times 10 nerves per inner hair cell. That's where this number roughly comes from. Okay. Back to the videos. What I just showed you. So neurons show a spontaneous firing. So if we record over time, we stick an electrode into an auditory nerve fiber, ANF, type 1, and we plot these spikes. These are individual action potentials. So if we would blow them up, we would have the picture from last week. But within 1 millisecond, they go up and down, and they undershoot a little bit. So that's quite accurate picture, actually, of what you see on an oscilloscope, because that's what you usually do in a real experiment. You stick an electrode into an auditory nerve fiber, and you have it on an oscilloscope, and you just listen to it as well. So you connect it to a loudspeaker, you hear the click, click, clicks, and what you see are, in fact, just these bup, 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 bup. Well, in real time, it's a lot faster, obviously. And if there's no stimulus, you still have some of these spontaneous spikes. If you present a stimulus now, I put those red bars always for a stimulus, starting here, ending there, you get more spikes. And if you have a very loud stimulus, you've got more spikes, even more. Okay. Yeah.
Is there any reason why we have spontaneous firing without having a stimulus?
Yes, I come to that. I hope I can explain that later on. It shifts your working point, effectively. it makes it more efficient because you can code positive as well as negative changes, if you like. And it is needed, this spontaneous activity, as is being shown by, if you don't have it, you suffer, especially with tinnitus. If you don't have spontaneous activity, for example, because you've got a neuroma which cuts off your auditory nerve and damages your auditory nerve, you obviously lose your hearing, but you will also get a tinnitus. And it is usually assumed that the spontaneous activity is there to create, if you like, a constant activity through the auditory nerve in the brain, which means that the brain knows that there's something coming in. And if that something isn't coming in, the brain makes up for it by increasing internal representation, which we perceive as a tinnitus. That's the evidence. Yes. Okay. Where does it go to in the cochlear nucleus? This is the cochlear nucleus. There's actually cochlear nucleus of a guinea pig, which I'm much more familiar with than the human cochlear nucleus. In the human, the DCN, that's the dorsal cochlear nucleus. So dorsal means further back, ventral further forward. They're actually separated. The DCN is a little bit further back there, and they're separated entities. In the guinea pig, and as far as I'm aware, all mammals, well, all experimental mammals, which are all small rodents usually, that's what we do experiments on. The VCN and the DCN are one entity. VCN, ventral cochlear nucleus, is the important one. Important in the sense of that's where the auditory nerve fibers all terminate. This is an electrode which is supposed to go into that area. I'm showing that to make clear what an electrophysiological experiment looks like. These names here, multi-oct, globe, spherical, don't put too much emphasis on that. This is very highly specialized information of what kind of brain cells you find in them. All of these neurons look slightly different if you look under the microscope, and they behave slightly different as well. And I want to give you an impression what a real experiment would look like and what we struggle against, if you like, to start, if you want to understand what's actually going on. So we're very much at the beginning of the understanding of how sounds are generally coded in them, mostly restricted by that we have only one electrode, but we've got here in the cochlear nucleus a few million nerves, and we need all of them, obviously, to perceive a sound, but we can only look at one of them at the same time. Okay, if we put an electrode into multipolar cell area here, we would observe something like that, for example, always in response to sound. So we've got no sound here, then we switch the sound on. Sound is quite often 50 millisecond pure tone. We use that, as we discussed in the tutorials, for some spurious reasons. Pure tones are not necessarily good example of natural sounds, but they're very useful to understand a linear system. And if we know that since we know that where we are on the basilar membrane exactly, a pure tone is useful because we know that we can characterize neurons very easily, because each of these neurons is only connected to part of a basilar membrane. Okay, what we would observe are these spikes, and as the tool of choice of what we usually do with that is what we do is what's called a post-stimulus time histogram. So a real experiment would have 250 runs. So we present the same stimulus 250 times, and to save space we plot each of these spikes just as one dot. So each of these dots is one spike observed within this 50 millisecond tone burst. This is a real experiment, takes about 2.5 minutes, and it's usually done to characterize the neuron. And you see that there are a lot more spikes. There are few spikes in the first 5 milliseconds, and then there are a lot more spikes in the beginning. And you see there's sort of strange structures in there. They actually have a meaning. Because, that's what I said, peri or post-stimulus time histogram, PSTH. If we actually use this, well, summarize over all of these runs, call them run, make a histogram out of that. So how many spikes happened at what point in time during the stimulus, we get to a point where we get to a graph like that. So as a function of time, 50 milliseconds the tone was on, you see that this is the number of spikes over all of these 250 repeats, or in other words, the probability that a spike happens. So the higher that is, the higher the probability that spikes actually happen in that time. And you see that a few things that are important. First of all, you see the spontaneous activity at the beginning. This red arrow indicates this is where we roughly start. So it's about 20 spikes or something anyway per second. And the neuron takes around 5 milliseconds to respond. Why is that? Because the sound actually needs some time to travel through the outer ear, middle ear, inner ear. Traveling wave takes some time to travel to the point of activity of the inner hair cell. Then the inner hair cell needs to change its membrane potential. The spike is elicited. It is going through the auditory nerve into this cell that we're actually observing here. And that all takes around 3, 4, 5 milliseconds. Less for the high frequencies, for the low frequencies, because the traveling wave is what takes most time, because it travels up the cochlea, and it takes then up to 5, 6 milliseconds for the low frequencies to get there. Anyway, once it's there, you get a massive burst, it's called an onset of spike activity, and then it gradually falls down. That's called adaptation, and then it falls rapidly down when the tone ends. And you see that there is a little gap behind it. That's because the neuron, after the tone is switched off, needs some time to recover, if you like. And it goes gradually back to its spontaneous activity, when it happily fires away spontaneously during the time of day, during no activity. Okay, there's a lot of information in there, some of which we understand, most of which we don't understand. Just to point out one important thing is, the most important change that you see, well, the change is the most important that you see, namely when you switch the sound on, the neuron goes wild. It will fire very rapidly for a short time, and then it gradually decreases. That's quite important because it reflects one of the most important perceptual things of our brain, of our hearing system, namely we're most sensitive to changes in sound. If I play a pure tone of 1000 Hertz in the background, if I would play it in this room, after about 2 minutes you wouldn't even hear it anymore. Because you have adapted to it, there is nothing important in that tone. It doesn't pose a threat, it's not interesting. But if I switch on, or if I even switch off that 1000 Hertz pure tone, if I switch it on, the neurons would do that. If I switch it off, the neurons would do that. You have a massive change. And this change is what's important. That's why language, well, that's a bit broad to say, but this is why language contains so many changes. We're not communicating with long whistles of the same frequency. We're communicating with rapidly changing frequencies and rapidly changing consonants on vowels, which have different frequencies and different times. We want to put as much change as possible because the change contains the information. And this probability, if you like, reflects exactly that, because the reason for that is that the neurons are responsive to the change of sound and not so much to just the being there of sound. Okay, let's start coding something, and we can code two things. One is frequency and the other one is amplitude. That's what the bits that we understand about neurons. Okay, that's why we present them in a lecture proudly. Okay, let's do a little experiment. Namely, we switch on this tone, same as before, 50 milliseconds. We do it 250 times, but we change the amplitude on a broad range, 20 decibel up to 80 decibel for some reason. Okay. Now, we know that we have spontaneous activity, and at this spontaneous activity there's not much going on in the neuron during the sound, so we would call that spontaneous. That's not above threshold. We usually define the neuron as having a threshold, and we would usually say the threshold is where this line, as a function of, I haven't said it, as a function of amplitude, it increases firing rate, and then it sustains its firing rate somewhere up here. That thing is called rate level function, because you've got a rate as a function of the level. And the threshold is usually defined where our rate is higher than our spontaneous activity plus the standard deviation. As simple as that. So we would say this threshold of this guy is somewhere over there. It's around 30 decibel. So at 30 decibel, this neuron knows, well, to 95% certainty, that there is a sound. Then we have an interesting range, because here between 30 and maybe up there, 50, standing a bit wrong, yeah, 50 decibel, it increases. So that's interesting, because it can change, it changes its firing rate as a function of the amplitude of the sound. And from there on, it doesn't change it very much. It actually drops a little bit. This drop is not typical, it's not what I want to point out. Imagine this as being, there's a lot of error in there, so there is not anything changing usually up there anymore, so it goes into saturation. It is not able, in other words, the spike rate doesn't tell you anything about the amplitude of the sound if you're above 50 decibel. So if you just had that one neuron, what could you say about a sound? You could say it is there once it's around 30 decibel, then you could say it gets louder up to 50 decibel, and then you can't say anything. So you have a dynamic range. I jumped that one and go back to that in a bit. You have a dynamic range. Maybe I do. You can only code amplitudes between 30 decibels and 50 decibels appropriately. And this leads us to what's called the dynamic range problem of neurons, which has not been understood for a long time. Okay. Connected to this dynamic range problem is, I do have to point it out, is the point that all of our neurons, these are auditory nerve fibers, not neurons, the auditory nerve fibers, the actual ones, have a different kind of spontaneous rate. Coming back to the question what it's for, it has been observed that we have 2 or maybe 3 different groups of spontaneous activity neurons, and about most of them, if we plot them as a histogram, how many neurons do we have with that spontaneous rate, or we put the threshold of the neuron versus the spontaneous rate, which is done here, you see that most of our neurons have a very high spontaneous rate, but a very low threshold. And the other, that's about 70% of our neurons, and the other 30% of our neurons have a very low spontaneous rate, so they're around 0, but they have a very high threshold. So you can either have one or the other. You can either have a high spont rate or a high threshold. But you usually want a low threshold, but apparently there are no neurons with a low threshold and a low spontaneous rate. And that's the crux of it. And that brings me to these are the 2 groups. Technically there's a 3rd group down here with a low spont rate, but I choose to ignore it and say just, okay, we've got low and high. That's much easier to remember. Right, dynamic range. We've got 2 types of these neurons that we described. We've got the ones that just showed you, good example, with a low spontaneous rate, so maybe 10 times per second it will respond without a sound, and it has a relatively high threshold, 30 to 40 decibel, and then it goes up for quite a while. Then we've got the ones with a high spontaneous rate, so maybe 30 to 40 spikes per second, but it has a lower threshold. So remember, the threshold is what we define where this goes up above the spontaneous rate. So we would maybe say this threshold of this one is over here, so somewhere between 20 and 30, and the threshold of that one is over here, somewhere between 30 and 40 maybe. So apparently what we buy with the high spontaneous rate is a lower threshold. And that makes sense, because let's plot the dynamic range. Dynamic range is small in these ones, you've got a very steep incline, and the dynamic range of these ones is large. Okay, what's the logic? The logic is the spontaneous rate seems to get the neuron to make it very sensitive. So they're hovering around the threshold, and even a little, tiny little change, even a random change, will make it go above threshold, will make it respond. That's what we see here. So they have a very high spontaneous rate, And a little change will get it over threshold, so we will have reached threshold immediately, and we will have a low threshold. So our absolute threshold of hearing at the very quiet frequencies will be determined only by our high spontaneous rate neurons. So this is the actual reason what the spontaneous rate is doing for us, because it gets us to be very, very sensitive. The problem why we not only have these high spontaneous rate ones is that if we did that, we only had a dynamic range up to something like 40 or 50 decibel. That's the dynamic range problem. So we need something else to explain higher dynamic range, because as we all know, we can differentiate amplitudes up to 100, 120 decibels, which is even further to the right here. So we need a dynamic range. We need some neurons at least that are still increasing much higher than that. And these are the low spontaneous rate neurons, which go up to much higher dynamic range. So the problem, or the solution of our dynamic range problem, is we've got several classes of neurons, depending on the spontaneous rate, and depending on the spontaneous rate, you've got a dynamic range, and you need neurons with higher threshold and larger dynamic range to cover 120 decibel. Okay, I could give you a minute with that now to digest all of that and press a button. So which one is false? 3 of them are correct, 1 is false. Auditory neurons respond to an increase in sound amplitude with a monotonous increase in firing rate. I think that's wrong. Neurons with a low spontaneous rate are responsible for the detection of a sound at hearing threshold. Is that right? Or is that wrong? Number 3, neurons with a low spontaneous rate are better suited for detecting the change of a sound. And number 4, spontaneous firing is a useful method to increase the neuron sensitivity. That one is correct. Okay. Number 1 is correct, number 4 is correct. I think everybody agrees on that. But which of these is wrong? Number 3, neurons with a low spont rate are better suited for detecting change of a sound. Go back. Low spont rate are better suited for a change in sound because they have a large dynamic range. That's correct. The bigger the dynamic range, the better you are in detecting a change of the sound, because you can code a difference between this amplitude and that amplitude, or between that one and that one. Where this one can't, because it's in saturation, just carries on firing up there, and you can't actually tell any change. Which means number 2 must be neurons with a low spont rate are responsible for the detection of a sound at hearing threshold. That's just the wrong way around, because hearing threshold is somewhere, well, I should maybe plot one at 0 decibel, because that's our actual hearing threshold. But in my lifetime, I have never found a neuron that actually starts responding at something, anything between below 20 decibel. I haven't found any literature on that either, but the common agreement is that people believe that you need a lot of them at the same time to actually make a threshold of 0 decibel. But if you do experiments, you'll find practically never anything below 20 decibel. But more to the point, these are the ones with the high spontaneous rate, because they are the most sensitive ones. Okay, we can put that all, one auditory nerve fiber, into a nice picture which shows us all of the information that we need to know about it, including the ones that I just gave you. And that's what's called the frequency threshold curve. So what we do here is we measure the neuron's activity in response to any sound between 260 Hz, 7 kHz. In fact, that could be the whole of the hearing range. We just choose to limit that a little bit because we know there's nothing happening in most of that time. And between 20 decibel and 80 decibel. And that gives us all the information to characterize the neuron where it sits on the basilar membrane, because its best frequency is where this pointy thing, this is what well, I should say, the more yellow there is, the more spikes we measure, obviously. So if we measure a 500 Hertz tone at 40 decibel, nothing will happen. If we go up and up and up in amplitude, at 70 decibel it will start responding. So it has a threshold at each of these different frequencies which is different. And it has a threshold which is lowest of all, which is somewhere here around 2 kilohertz, and that's the lowest threshold, and that's what's called the best frequency. So that's the threshold, the frequency where it responds best at lowest threshold, which means we know now where it sits on the basilar membrane, namely exactly at the 2 kilohertz site, where the traveling wave has its peak at 2 kilohertz. Because, nothing magical about that, if you take this curve, it looks very much like the filter shapes that we talked about all the time. In fact, this is where the whole idea of filter shapes comes from measurements, electrophysiological measurements of neurons, of auditory neurons, because they show you this filter. This is the auditory filter. This is one of our auditory filters, of one hair cell, or one auditory nerve fiber, in fact, of which we have 30,000. But this is all that we need to know, because we have a best frequency. We know its threshold is around 30 decibel here, at the best frequency. And we know that the filter shape indicates something about its goodness, its quality, what it's called. We define quality later on. That's basically the width of this curve. We also know that at higher frequencies, nothing happens. That's due to the traveling wave. It just breaks down at higher frequencies, and you can't get anything out of this hair cell if you go up to higher frequencies. But at lower frequencies, you see that they are becoming a bit wider. Yeah.
Question about the dynamic range of the nerves. Wasn't it supposed that they only have a maximum dynamic range of 30 dB?
Yes.
So the type 2 that we said that have an extended
Dynamic range.
one, it doesn't really work, no? Because what makes the amplification is the nonlinearity of the cochlea, or no?
No. The question is, has it got anything to do with the nonlinearity of the cochlea? Yes and no. Going back to the dynamic range, maybe we should discuss that later on. This is a working cochlea. This is not a dead animal or anything, but this is an actual working cochlea, so that includes all of the dynamic and non-linear processes of the cochlea and everything. That's not, well, what we observe as the dynamic range of the neuron, but this is a function, of course, of everything that happens before. So the cochlea, with its active amplification, has increased the dynamic range, and all of that is not a function of the neuron, but it's just reflecting the cochlea. So, the frequency threshold curve displays the extent in frequency and amplitude in which neurons respond. It shows the best frequency, sometimes all the characteristic frequency. Best frequency is British, characteristic frequency is American, if you read the literature, so we call it best frequency. That is the frequency at which the neuron responds at the lowest amplitude. It shows us the threshold of the neuron. That is the amplitude at which the auditory nerve fiber responds at its best frequency. It shows us the asymmetric towards low frequencies, so it goes up in a big arch at low frequencies. It's very steep at high frequencies. That reflects the traveling wave, and it's the inverse of the auditory filter. And since we ran out of time, I'll leave it here and catch up with the rest of the lecture next week. Talk about the filter.